Hey everyone, my name is Vatsal Sivaratri. I'm a sophomore and I live in Ashburn, Virginia. I'm very excited to present my research on EEG biomarkers to characterize and predict Parkinson's disease using a novel algorithm and a deep learning model. I worked on this project with Kevin. I chose to take upon this research because it aligns strongly with my passion for machine learning in bioinformatics and health sciences. I strongly believe that research in this field is indispensable for the well-being of humanity. Without further ado, let's get into it. Parkinson's disease, or PD, is a prevalent and challenging neurodegenerative disease that hinders a person's motor control, causing extreme discomfort and difficulty in day-to-day activities. With there being no current cure for this disease, early intervention is the best way to improve patient outcomes. Current methods to diagnose this disease rely on examinations performed by overqualified neurologists through examining medical history and symptoms. Unfortunately, this approach has proven to be valuable, with the misdiagnosis rates ranging anywhere from 10 to 20%. Furthermore, this method lacks early diagnosis, with most patients having to wait months to years before symptoms show up. EEGs are diagnostic tests that measure the general electrical activity of the brain using small electrodes attached to the scalp. The data collected from this technology can be crystallized to making accurate inferences about neural activity and differentiate between neurological conditions. The cycle-by-cycle algorithm, or Bycycle, is a novel approach for analyzing neural oscillations in the time domain. It segments the signal into individual cycles and analyzes them separately. The algorithm's advantage over conventional methods is that it allows for a more direct and accurate characterization in the time domain rather than the frequency domain. The thesis of this project was to verify the applicability of this new algorithm for EEGs in PD. Can we rely on Bycycle to understand and hopefully prognose this disease, ultimately improving the lives of millions around the world? Through my project, I wish to determine exactly this. Let's get into it. To conduct this study, I needed a sufficient dataset. When looking for a dataset, I kept in mind to pick one which had diversity, good quality data, and sufficiently long readings. I eventually selected a UC San Diego dataset for resting state EEG readings. The dataset contained 31 patients, 15 with Parkinson's and 16 healthy controls, with a roughly equal gender split for each population. The recording was 3 minutes long, with a frequency of 512 Hz, leaving me with ample data for the analysis. To determine if Bycycle can effectively differentiate PD, I conducted two sample t-tests for differences in population means. I did this by downloading and extracting the data using the MNE library. Specific channels were selected that pertain to the prefrontal cortex, an area of the brain that relates to motor control and is affected by PD. These 12 channels were run through the algorithm individually and averaged. A table of 24 features, including voltage, amplitude, period, and symmetry was generated. These features were averaged among their respective populations, and 24 t-tests were conducted for each feature. As seen in the table, there is a statistically significant difference in nearly half of the features computed. To create a proof of concept upfront of my findings, I implemented a neural network to label features as PD or control. I chose to use a sequential model. Between each layer, the model uses batch normalization and dropout, which are techniques to prevent overfitting in the model. I cleaned and filtered the data using MNE and ran the model on the same channels of interest. I then truncated the data and preprocessed it to fit the shape of the model. After iterating through 50 epochs, I achieved an accuracy of 96.15%, with the model reaching 100% on some epochs. Here are some graphs of the model's performance. The results of the study bring some very important conclusions for future study. Firstly, a significant difference between over half the features implies that Bycycle can and should be used as a diagnostic tool for PD and to further understand the disease. Secondly, the Vol features showed extremely low p-values from the test, reaffirming a great difference in overall neural activity. Finally, the model was able to classify the data with 96% accuracy, meaning that with some fine-tuning, we can create a viable, cost-effective solution to accurately detect PD earlier. Future research that can be done to build upon this would be to investigate the use of Bycycle with other neurophysiological data that models PD. We can also explore another part of the brain, such as the basal ganglia, another region known to be affected by PD. Finally, the potential of this technology doesn't just have to be restricted to PD, but can be implemented to other neurological conditions too, such as Alzheimer's and sclerosis. I'd like to thank everyone so much for your time, and the floor is open to any questions you might have.